do it a few times.
This is so embarrassing.
It's different when you're recording yourself.
Yes, it is. It's more intimidating. Alright, thank you for coming to my 10-minute presentation. Today I'm going to talk to you guys about Unionids, or more widely known as freshwater mussels. Now, the reasons why we study freshwater mussels in the first place is because they're highly diverse throughout the United States and all of North America in general. But not only that, we study them because they're highly endangered relative to the other different kinds of animals in the world. Now, our objective for my research, my summer research, is that we're going to look at the current status of freshwater mussels in a stream in Hastings, Michigan, Cedar Creek, and we're also going to try and attempt to understand the relationship between freshwater mussels and macroinvertebrates. Now, there's different influences in the decreasing population of Unionids in the United States, and under the umbrella term anthropogenic influences, which means human-caused, the four main reasons why the population of Unionids have decreased are the button industry, dam construction and destruction, poor water quality, and invasive species. Now, the button industry took place 1700s all the way up until about 1950. Freshwater mussels, the other side of their shell is really shiny and really pretty. So up until about the 1950s, the streams, rivers, and lakes were dredged for freshwater mussels, not only for a food source, but to use their shells for buttons. It wasn't until 1950 that plastics became readily available to manufacture and create the buttons, and that was when freshwater mussels became widely known and widely it became a public concern about their decrease in population. Dam construction and destruction is a modern concern for freshwater mussels because dams are becoming more and more expired, which means they need to be either rebuilt or taken out. Now, you might not think taking out a dam is as detrimental as building a dam, but it actually, in the short term, it can actually completely desiccate an entire population of Unionids within a stream column. Long term, it's good for the stream, but short term, you can completely wipe out the entire population. Now, poor water quality, there's direct point and non-point pollution, which means you're either getting pollution straight from a factory and you can pinpoint exactly where that pollution is coming from, and there's non-point pollution that has to do with runoff, which is when rain runs off into the closest body of water and brings in contaminants like animal feces and other different kinds of contaminants that don't belong in the freshwater column. Invasive species is also one of the most modern concerns for the decreasing Unionid population. Zebra mussels and quagga mussels are invasive species out of Europe. In the 1980s, they were introduced from stored water within the ships bringing in different cargo into Lake Michigan and all of the Great Lakes. These mussels are invasive, for one, because they are not native to here, and for two, they take over anything. They can plant themselves onto any flat surface, even on top of native mussel species. They are not like Unionids. They do not have the certain life history characteristics that Unionids do, which I will explain in a second. But zebra mussels and quagga mussels attach themselves to anything and cause millions and millions of dollars in damages to harbors, to boats. They attach themselves to literally anything. And they can take over the masses of the stream column. Now, Unionids are a family of freshwater mussels that have a unique life history characteristic. Unionids separate themselves from the rest of freshwater mussels because they require what's called a host fish, which means that they need this certain kind of species of fish to carry on their juveniles. So what happens is a host fish, this is a lure, by the way, in this video. Some Unionid species have these. They mimic the fish's prey, The fish bites on the prey thinking that it's food. The mussel senses the bite and ejects its glaucidia, or you can think of it as like a zygote, thousands upon thousands of these glaucidia into the mouth in the water column of the host fish, and then the glaucidia attach to the gills of the fish, filtering out the nutrients and different kinds of other nutrients coming in and out of the gills until they're old enough to do it on their own. Now, macroinvertebrates are really important for different reasons. One of them being that benthic macroinvertebrates differ in their sensitivity to pollution. So the picture on the right is what's called a water penny. It's one of the most sensitive macroinvertebrates to pollution in the United States. So if this is present in the stream column, you can make a relative conclusion that it's a healthy stream. Also, benthic macroinvertebrates provide information about the quality of a stream over a long period of time. So yes, they do move. Yes, they can move from stream to stream. But if there's a high diversity of different kinds of macroinvertebrates and there's a good mixture of high tolerance and low tolerance of pollution, that also gives you a good estimation of stream health. Also, benthic macroinvertebrates are relatively easy to collect versus mussels or other different kinds of fish. All you really need is a partner and a kick net or a Hess sampler, which I'll show you guys in a minute. And not last but not least, benthic macroinvertebrates are an important part of the community of life within the stream and outside the stream. Now, when I say that, I mean that a stream collects leaf litter and also builds up algae on the bottom, or the benthic layer of the stream. So the macroinvertebrates eat that, and then the fish eat the macroinvertebrates, then the birds eat the fish, and raccoons eat the fish, and there's this chain, this food chain that macroinvertebrates, without them, that interaction between the riparian zone, which is the land outside of the stream, and the stream, that interaction wouldn't be there without the macroinvertebrates. Now, my study site is in Hastings, Michigan, which is about an hour and 15 minutes south of Grand Rapids. And the reason why we chose this site is because there was a previous study done 3 years ago about the current status of Unionids present in that stream. And a big thing about conservation of anything in general is keeping track of the populations, of the known populations within a certain site. At this site, we are doing sampling over a 200-meter reach within the stream. Also, we are doing 18 different sampling sites, which equals 36 square meters of sampling for mussels and macroinvertebrates. Now, my methods include sampling of the macroinvertebrates, which includes preserving them to analyze them in the lab, and visually identifying the mussels and counting the populations and then replanting them into the ground. The picture on the left is showing myself using what's called a Hess net sampler, which is a bottomless bucket that has a net in the front, which is facing upstream, so the water's going this way, which is making water go through the bucket and through this net right here. So, what is this, right? So the bottomless bucket has an opening about of that. You mix up the substrate, clear off the rocks, get all the insects and everything out of that column, and let the stream push them into this container right here, where I'll collect them, put them in a bottle of ethanol, and then analyze them in the lab. The pictures on the right show me visually identifying and sampling Unionids. There's no special equipment to do this, but because they are endangered, the Department of Natural Resources only limits you to visually collecting and taking home the dead shells versus the live shells. Now, functional feeding groups are what ecologists over time have categorized the macroinvertebrates in 4 different groups, and the groups indicate how and what they eat. So it's their eating behavior and what they consume. Shredders, gatherers, scrapers, and predators are the four main functional feeding groups. Shredders consume seed palm or coarse particulate organic matter. Gatherers, which include filter and collectors, consume F-palm or fine particulate organic matter. Scrapers consume periphyton and algae, which includes diatoms and other green algae within the water column. And predators obviously consume prey. Coarse particulate organic matter includes leaf litter, large woody debris, stuff that you can visually see with your eye. Gatherers will either create these nets out of silk within the water column, and it could be anything from smaller pieces of the wood within the water column or even feces. Scrapers, obviously the algae and the predators of prey. Some preliminary results that I have done include using these functional feeding groups mathematically to estimate stream health. So for example, after combining scrapers to predators and collectors, we found that the stream was heterotrophic, which means they get their energy and food sources from outside of the stream. So in layman's terms, there's not enough algae and not enough inside sources to feed everything that lives within that water column. Also, combining scrapers, filter feeders, shredders, and gatherers, we calculated that the channel stability is high with numerous attachment sites for macroinvertebrates. So what that means, there's a lot of, more layman's terms, but there's a lot of caddisflies and a lot of other insects that have silk nets and that use these different kinds of nets within the water column that are attached to all the different substrates on the bottom to collect food. Now the graph above shows the correlation between the population of macroinvertebrates, which is on the left in blue, to Unionids, which is on the right and it's in red. Now, a whole point of our objectives for this research is to see the correlation between the populations. So for the 5 sites that I've analyzed, you can see relatively they all increase when the Unionid population is high, and then they both start to decrease when the Unionid populations become lower. So in conclusion, still need to work on that. We still have further research to do, and thank you guys for coming out. And if you guys have any